In this meeting, I presented results from our single-cell RNA sequencing cohort of bone marrow and peripheral blood immune cells from patients with high-risk smoldering multiple myeloma enrolled in a phase 2 clinical trial of Elotuzumab, Lenalidomide, and Dexamethasone. We found that there are significant changes in the immune cell composition of patients with smoldering multiple myeloma compared to healthy donors. We saw increases in CD4 positive T cells, granzyme B expressing cytotoxic T cells and CD56 DIM NK cells, as well as decreases in CD14 positive monocytes with a switch to CD16 monocytes and decreases in plasmacytoid dendritic cells. But the bigger question is, a lot of these changes we already knew before. How can we finally leverage these changes to actually prognosticate for our patients and understand who is going to progress, who is going to respond to therapy, especially immunotherapy? So first we hypothesized that the similarity of a patient's immune cell composition in the bone marrow to that of a panel of healthy donors may actually be prognostically relevant. And so indeed, what we found was that patients who are least normal-like at baseline, or patients who we called reactive, are actually the ones that have significantly longer progression-free survival upon treatment. And not only that, but the normalization scores increased on average from baseline to end of treatment, which is important because there's this whole conversation about immune reconstitution post-therapy. And so we called it post-therapy immune normalization, or PIN, and we determined that based on the change in normalization scores from baseline to end of treatment. And we saw that patients who achieve post-therapy immune normalization have longer progression-free survival. And perhaps the most important finding of the study was that peripheral blood samples actually reflect the immune dysregulation we normally recover in the bone marrow. And this is really exciting because it means that in the future we may be able to profile our patient's immune state with a simple blood draw. And I think one of the biggest challenges in bringing immune profiling to patients is exactly that, is understanding whether we can do it in the blood and therefore do it regularly, do it often, and really improve our prognostic models. We already have great models. They're largely clinical. We have lots of papers showing that genomic biomarkers can further improve them. So now is the time for immune biomarkers to also enter into that equation, and I think that we can really do better with our prognostication.